Bonjour. Hello. I'm Alexandre Brevern, and today I will share with you the work I have done with my collaborator Aline Flock and Stefan Telechea. Our research deals with the contribution of bioinformatics to blood transfusion with two aspects: 3D intraprotein interaction studies and a database. First, I would like to thank the committee for the opportunity to present the works of our laboratories to 3DSIG in Lyon 2021. Thank you. I will begin with the transfusion. In fact, more than 3 centuries ago, we have the first documented blood transfusion fulfilled to a human being. It was done by the personal physician of King Louis XIV. We have a poor young boy, 15 years old, that was suffering from a lot of bleeding made by leech, a very classical treatment at this moment, and he had a high fever. So Dr. Denis decided to transfuse the blood from a sheep here on the picture to this poor little young man. And very surprisingly, he survived. Today, of course, it's more sophisticated. And we have all seen different pictures asking for us to provide blood for transfusion. But we all know that there is still some issues with transfusion, and these issues are not made by vampires, but by something a little bit more dangerous: the blood systems. The most known one is the ABO, that is made by sugar on cell surface of the proteins of the red blood cells. And vampires does not exist, ABO exists. The blood groups, what it is exactly, and what happens with the immunization. An individual devoid of an antigen, so he is negative to the antigen, via transfusion or pregnancy is exposed to this antigen. We have, for the antigen, a specific immunogenicity. There is, of course, individual factors for each one. Clinical context, if there is other type of disease, and the prevalence of antigens, depending on the environment. And so, sadly, sometimes you have an alloimmunization events, meaning you have antibodies produced by the human bodies against these antigens, and it can create hemolytical consequence for post-transfusion hemolysis, or more specifically, when you have pregnancy for the fetus or the newborn. At these days, we have 43 blood group systems that have been exposed with 345 antigens. It evolves. Most of these antigens are present on proteins on the surface of red blood cells. There are very diverse categories, some with sugar, some with proteins, some with sugar with proteins, and they are highly complex. Of course, there is a lot of tests when you have transfusions that must be done for ABO and RH, rhesus RHD systems. After ABO, RH is the most problematic one, and also is a more complex, because you have seen ABO, we have 4 groups. With RH, we will see it's quite more complicated, sadly. So what is a RH group? It's very simple. At the beginning, you have 2 paralogous genes, RHD, RHCE. They are very close in the genome. They are highly similar, 92% of sequence identity in terms of amino acid. So they encode for 2 proteins, 2 transmembrane proteins, to be more precise, and they define 5 main antigens for RHD and RHCE. We are mainly interested into the most problematic one, RHD. So, the story of this works. And I will firstly present myself. So I'm Alexandre Brevern. I am a senior researcher in Medical Research Institute INSERM. I'm the head of the bioinformatics team ECCB, and I'm a structural bioinformatician, and I've been specialized in different blood groups, for instance, the Duffy, or the very specific one on integrin alpha 2b beta 3 on the platelet, so only blood group that is not on red blood cells. My main collaborator on this project is Dr. Aline Flock from EFS, with Christophe Tournamil and Franz Pirenne, the head of the team. They are Aline and Franz are medical doctors specialized in transfusion and of course RH. And now Aline is also specialized in bioinformatics. Okay. So some years ago, our collaboration began with very simple questions. Christophe Tonamil arrived and tell me, I have a new RH variant associated to transfusion events. It's a partial D. Okay, that's the terminology in transfusion. So he asked me what happens, in fact, in 3D. What is the impact of this mutation? So I have proposed structural models. For that, of course, going from the reference of RHD sequence, I have searched in different databases, and especially, of course, the protein data bank with different tools, PsyBlast, HMM, and others. And PsyBlast, for instance, provided me a very first potential structural templates, good coverage, 32% of sequence identity, and a second one, a partial one, only the beginning. But in fact, when we were looking at the two structural templates, they were highly similar, and also, in fact, you can align them with very good accuracy. So we have used the two structural templates to do comparative modeling. So here we have done this alignment, and we have used Modeler to do the comparative modeling. To select the best, potentially best, of course, structural models, we have not used the DOP score because it's not a globular protein, but we have used dedicated tools. For instance, the one we have developed inside the lab, one is called Maiden, another one is called HPM for hybrid protein models. And we have gone to models that we were very happy with it, and it is this model. The interest is not, of course, to propose simply model, but to try to use it. So we have done the punctual mutation using a spiral methodologies developed by Roland Dunbrack, and we have looked which type of interactions were maintained or not between the structural models we have proposed and, of course, the different variants. And what is very interesting is that, in fact, the original amino acids, the phenylalanine in position 223, made a lot of interaction, and the variants, it's very simple. It's highly drastic. You lose a lot of interaction, and you lose a lot of interaction with a lot of helices around this position. So here we have an hypothesis of protein destabilization locally, potentially, that create cryptic epitope that are implicated into the immunization events. Okay. So it was, of course, a very simple question, and it has grown to a more complex question because, in fact, RH is not a solo player. It's a trimer with another protein called RHAG. It's coming from the same family, of course. And is it possible to propose a trimer when we don't know the stoichiometry? And is it possible to use it to go further? So here again, we have gone to propose a structural models, a more refined one, using as a structural template not only one protein, but directly a trimer. And after refinement, using a little bit the same type of approaches, we have selected this structural model for the trimer with 2 RHG and 1 RHD. There is information why we have used this one. So here, same type of questions. We have wild type. We do punctual mutation with squirrel, and we look at the interaction using approaches to determine which type of interaction are gained or lost, locally or within the environment. So we take a little bit less than 20 different variants. Some are important for immunization risk, some are frequent but with a weak impact. In green, you have the less frequent, the very frequent, sorry, but with a weak impact. And on this plot, we have, on the x-axis, the number of interactions that have been modified. So here, 0 interactions have been modified. Here, 20 interactions have been modified. And on the y-axis, that's the number of interaction between the domains, meaning between the helices that have been modified. So here, nothing, no helices change. Here, going to 4 interaction with 4 different helices that have been modified. And so what is interesting, and I come back to the I mean the typology of each variant is in green. It's frequent but with weak impact. And we have, in fact, a very limited number of interactions that have been modified, and at most with one other helix. And when we have the very important variants that have a high elimination risk, we see that there is a lot of interactions that have been modified and also going with two or more helices. So it's a very interesting result. From this analysis, we have already seen that we have a better definition of extracellular domain of RHD. And it's very interesting because medical doctors are using the same representation in 2D like this one since 30 years at a time where we have no information about topology. So it's a better definition, and even for medical doctors, it's interesting to have our own definition. Second point, by analysis of these intraprotein interactions, we have seen that we have a nice correlation between the impact on immunizations and the change of intraprotein interactions. So it's very interesting result. During this collaboration, I was very surprised to have a very difficult access to proper information on RH variants. There is RH databases, but in fact, it's more HTML pages. You cannot do any request, and it's not properly maintained in some times. Okay, but as we are bioinformaticians, we have decided to develop a specific database, not databank, as it is these days. So here comes the third guy, Stéphane Tellecea, former postdoc in my lab. He's an assistant professor at Nantes University. He's a very good structural bioinformatician, but also very good in informatics. He had provided the RESPIR database, where you have a large number of information about proteins of the red blood cells. So, first point, Dr. Aline Flock, she has done an amazing literature curation, reading more than 500 of articles and conference abstracts. And she has put everything into a large CSV file with everything that is in some way copy-paste, but also reanalyzed. So meaning it's really an impressive work, a human impressive work. And here, that's a representation of a very small part about this file. And below, made by Stéphane Tellecea, the real table behind the database. We have something like close to 60,000 cells containing between 1 and 70 data. More than 25% of them are completed. So it's really a big systems of information. And here you have a very simple presentation of RH reference database. You can ask for directly your variants of interest. You can ask for a different type of informations in a very simple way, even for medical doctors, but also for researchers. So, meaning we have provided also statistics, different type of documentations that are very precise. So here you have the you have a very classical variant page where you have all the information that can be seen here. And as there is a lot of information, we have decided to put buttons so you can extend the information to have everything you want. It was published this year in Transfusion Medical Review, and it's very well recognized by the medical community and the scientific society for transfusion. So it's very good, and we are very happy for that. It's very useful. So to conclude, I would like to share with you some unpublished works, because of course we have a lot of information. We have structural models, so we have decided to go for molecular dynamics of this RHD, RHAG trimers. And here you have a representation about results from the MDs on the RHD. Of course, extracellular and intracellular loops shows interesting movement, but already we see there is some strange things within alpha helices here and here. And we are using for that innovative approach based on protein blocks that we have developed in my lab. So to conclude, I will say that we have a very fruitful collaboration between structural bioinformaticians and medical doctor, which is quite unusual. And moreover, I can say that Dr. Aline Flock, she is a medical doctor, is perhaps the only medical doctor that I know that can do really excellent molecular dynamics. So it's really a very successful collaboration. We have shown that structural bioinformatics can change the view of proteins analyzed since decades by specialists. And it's very important because it can help them a lot. Moreover, we have shown that there is potential links between polymorphism and transfusion complications through structural models. That was our main goal, and it's successful at the end. And moreover, from all those discussions, we have proposed a new reference database where you have 3 times more information than in the current database. And that is really something where you can ask for something, you can do some query, which is not possible at this moment on the other database. So it's really a great success. So we are very happy with this collaboration. And I would like to thank you for your attention, and I will be happy to take any type of questions.